Hi everyone, good evening, very good evening to all. So welcome to Rapid Revision Program NEET 2025. So again welcome to the zoology class now. So in the previous videos we had been discussing some important topics that related to the NEET 2025. Most weightage chapters we are covering. In the last class we had discussed about the breathing and exchange of the gases. That is one of the most important chapter in human physiology. So now in this class we will be discussing with body fluids and circulation chapter. Body fluids and circulation chapter we will be discussing. This is the most important chapter. From the chapter you will get 3 questions or 2 questions sometimes. 2 question or 3 question you will get in the examination, right? So most weightage chapter, all human physiology chapters are very important, right? So in this class I will be explaining the NCERT line to line that will help you to uh, that will help you that will helpful for you to revise the chapter in 1 hour. In 1 hour the entire chapter will be revised, and that will be helpful for you for NEET 2025 exam, right? So body fluid and circulation chapter. So I will be covering that NCERT line to line and how they will ask you question from each sentence I will be explaining in this class. Okay, right. Body fluids and circulation chapter. Before going to this, so the introduction part in NCERT book, the introduction page of the NCERT book, they have mentioned that all the living cells are continuously supplied with nutrition, oxygen and essential substances, and also the waste material or harmful substances which are produced in the body should be eliminated. Okay, so this statement that is given in NCERT book, that is very important. What is that statement? States for healthy functioning of the tissues, healthy functioning of tissues. Okay, so the tissues which are present in the living organism, for healthy functioning of the tissues, the living organism continuously supplied, we have to provide continuously, oxygen should be provided, nutrients should be provided, and other essential substances, other essential substances. So continuously these are the material or component continuously we have to supply, right? For all living organism, very important. All living organisms. Okay, for healthy functioning of the tissues that present in all living organism, continuous supply of these materials are very much needed. Not only the supply of this material and the waste material which are produced, example carbon dioxide, harmful material, and other waste material should be eliminated from the body. Because of the metabolism inside our body or inside the cell, so waste materials will be produced. That waste material will disturb the equilibrium of the body, or homeostasis of the body will be disturbed by the that waste material if they present inside the body only. So that's why continuous supply of the material should be needed, and also the waste material which are produced during the metabolic reaction inside the living tissues also should be eliminated. Okay, so for this, for the continuous supply of this material to the cells and also for elimination of waste material from the cells, we required a system, an efficient mechanism is required. We required an efficient mechanism, right? So an efficient mechanism is required. an efficient mechanism is required. So what is that efficient mechanism that can help us to supply these material to the cell and eliminate these material from the cell? What is that efficient mechanism? These efficient mechanism, in case of the simple organism, the efficient mechanism in case of the simple organisms like sponges and ciliates. Okay, these are the simple organism. In this animal, the water current, the water from outside enter into the body cavities, right? And this water in the body cavity, and through the body cavity, the cells or the cells which present in the tissue, they getting nutrition and oxygen and other material. And also these water which is coming out and enter into the coelom or body cavity, from that body cavity it is going out, right? So when that water is going out, it is also taking the waste material from the body cells. So this is the mechanism that helps in transportation of these material to the cell, and waste materials are away from the cells. This is the mechanism that present in simple organism. But in complex organisms, including humans, okay, in complex organism including humans, there are two special type of liquids are present. There are two special type of liquids, that is one blood and other is lymph. Okay, so in complex organism there is a special type of liquids are present in their bodies. That liquids, mostly blood is the common liquid that present in our body, that helps in transmission of these components to the cells and also away from the cells for elimination, right? So lymph is also helps in transportation of certain substances. Right? Now, these efficient mechanism is not same. That efficient mechanism which is helping in transportation of these material to the cell and elimination of these material from the cell, right, is different. The different group of animals have evolved different types of mechanism for elimination of these components. So this is very, very important. Okay, I will write here different group of animals. Different group of animals have evolved different types of mechanisms. Okay, so from these area, so how they will ask you question, I will tell you. So don't miss that important points. So that is given in the introduction part of the body fluid and circulation. This is also very, very important. So the first, how they will ask you question is, all the living cells are provided with oxygen, nutrient, other essential substances. This is the right statement, right? For healthy functioning of the tissue, in the living organism, which of the following is required? Continuous supply of these materials is required. At the same time, elimination of these material also should be required. Okay, so they will ask you question, statement based question, correct statement, incorrect statement, identify incorrect statement, identify correct statement in the given, right? So that you should able to know that. And so based on these also they will ask you question. different group of animals have evolved same kind of mechanism for transportation of this material to the cells, away from the cells, they will ask you. If same means that is the wrong statement. So different group of animals have evolved different types of mechanism. Example, lower organisms or simple organism like sponges and coelenterata, you studied that water transport, water from outside along with the food and oxygen going to the body, and after that it collecting waste material from the body, eliminating outside. But whereas in complex organism there are certain liquids are present or specialized fluids are present, we are calling them blood and lymph, right? So this is important. Okay, so I hope that will help you understood that first page of the NCRT very right. So don't miss that, the first page of NCRT also you read line to line. Next, so blood, the liquid is the blood, special connective tissue, special connective tissue. So it is made up of with fluid matrix present. Okay, fluid matrix. Suppose this is blood vessel. So this is the liquid or blood. Now you can see here all extracellular matrix, that is the lymph and blood. Intracellular matrix, cytoplasmic fluid. Sorry. This is intracellular matrix that present inside the cell, cytoplasmic fluid. Outside the cell you can see these are all WBC, RBC. Now the liquid which is present outside the cells of RBC and WBC. So the liquid portion of the blood we are calling extracellular fluid, that is blood and lymph. So blood is a specialized connective tissue. We know that there are cartilage, bone and blood. These are the specialized connective tissue. Important. Right. So blood, composition of the blood. The blood is formed by plasma, the liquid part, and formed element 45%. Right. So 55% plasma and 45% are formed elements. So plasma is a straw color viscous fluid, okay, and it consisting 90 to 92% water, very important, 6 to 8% proteins, 6 to 8% protein, 90 to 90% of the plasma will be water. So your question they will ask you, the most abundant component of the plasma is, water, right? What is the total percentage of proteins in plasma? 6 to 8% of proteins are present in plasma. And they will ask you which of the following is not correct related to the plasma. Plasma is about to 55% is the right statement. It is a straw color, right statement. Viscous fluid, right statement, right? So they will ask you question, which of the following is not related to the plasma? Next. So proteins, fibrinogen, this is the protein that present in blood, plasma, fibrinogen, albumin. So fibrinogen is a blood clotting protein that helps in coagulation or clotting of blood. And osmotic balance is maintained by the albumin protein. So globulin protein maintain defense mechanism because globulin protein produce army of proteins that is released by or produced by globulins. We are calling antibodies. So these antibodies, they can fight against disease causing microorganism or pathogen. So So these are the proteins of the plasma involved in defense mechanism. Question important. Which of the following protein are involved in defense mechanism? So globulins. So osmotic balance of the blood maintained by the albumin. So albumin, how it maintain osmotic balance, just I will give you some clarity on this. You can see. Now you can see this is white color. This is the albumin protein which present inside the blood vessel. Inside the blood or inside the plasma, this is albumin protein. Albumin protein. So albumin actually negatively charged protein. Negatively charged protein. So it attract water molecules. Water is a positive charge. Water is a positive charge. So the water molecules are attracted inside the plasma. So now it maintains the balance, osmotic pressure. The blood pressure is maintained by the albumin. So imagine if there is no albumin in the blood, what happens? The water that present in the blood is again it is moving into surrounding. If there is no albumin protein in the blood, so the water that present in the blood, right, that is in plasma, will moves into surrounding tissue, right? So the surrounding tissue will be swelling or inflammation will be there. That inflammation we are calling edema, right? So lack of which protein that lead to inflammation of the body fluid, inflammation in the body parts, or swelling of the body part, that is albumin. Important. So other component that present in plasma, minerals, sodium, chloride ions, magnesium ions, bicarbonate ion, calcium ion. These are the ions that present in plasma. Glucose, amino acid, lipids also present in transit form. Transit means they can circulate from one place to other place, or their concentration will be changing. Okay, they can circulate from one place to another place through the plasma, or their concentration will be changing. Okay, so that's why we are calling them their transient form, right? Plasma, if you remove the clotting factor from plasma, the remaining component we are calling serum. So sometime they will ask you question, among the following, which of the following will not be clotting? Which of the following will not be clotting? So they will give you blood and plasma, serum. So serum is the answer because serum doesn't have any clotting factor, so there is no clotting takes place, right? So serum. Okay, this is about plasma. Next one. So RBC, formed elements. So RBC, WBC and platelets. These three together collectively known as formed elements. All these formed elements are formed in bone marrow, right? So the mechanism of formation of the WBC and RBC both are same, but formation of the platelet mechanism is different. Okay, so the formation of the platelet mechanism is different compared to WBC and RBC, right? So platelet formation is by the fragmentation, breaking of the cell, but here they are produced from the stem cells. Here one cell is broken into small pieces, but these are one cell develop into other cell. Okay, now so RBC, next we will discuss about RBC, WBC and platelets. So RBC red in color, red blood corpuscle, because of a complex hemoglobin protein present, very important complex. Hemoglobin is a complex protein which contain iron. Question important. Iron containing complex protein is hemoglobin that present in RBC. Because of this hemoglobin protein, they are in red color. Okay, red color, iron containing pigment present. So the significant role of this hemoglobin pigment helps in transportation of gases, oxygen and carbon dioxide transportation. transportation of gases with the help of these complex protein. Okay. Healthy individual 100 ml of blood contain 12 to 16 grams of hemoglobin. In 100 ml of blood, 12 to 16 grams of hemoglobin present. Okay, 12 to 16 grams. So the lifespan of the RBC 120 days. The lifespan of the RBC 120 days. After 120 days, the destruction of RBC will take place in spleen and liver. Only 90% of RBC destruction takes place in spleen. Only 10% RBC destruction takes place in liver. The 10% RBC, those were destructing in liver, converted into bilirubin and bilipigment. Bilirubin, biliverdin pigments. Okay. So 90% of RBC destruction will take place in spleen. Right. So the spleen is called graveyard of the RBC. Graveyard of RBC. So the location or the place where RBC destruction is taking place, that area or the place we are calling graveyard of RBC. Now the production of RBC taking place in bone marrow in adult. Bone marrow is the site where production of RBC takes place. So it is called cradle of RBC. Cradle of RBC, production of RBC in adult takes place in bone marrow. Liver and spleen also helps in production of RBC when embryonic stage. Liver and spleen both also helps in production of RBC, but in embryonic stage. So one more also, you remember just yolk sac. Yolk sac also helps in production of RBC in early embryonic stage. So embryonic and early embryonic stage, yolk sac and liver and spleen is the site for the RBC formation. But after birth in adult, bone marrow is the main site for the production of RBC. So it is called cradle of RBC. So healthy man contain 5 to 5 million RBC per cubic milliliter of blood. 5 to 5.5 millions of RBC. And RBC are enucleated. No nucleus, Rolex or dumbbell shape. No nucleus, Rolex or dumbbell shape, right? So most abundant cells of the blood are RBC and they are enucleated. Most of the mammals, almost all mammals are without RBC, RBC without nucleus. Nucleus absent. All mammals, almost important, almost all mammals are without nucleus. The RBC of the most of the mammal, that means possibility 90% or majority without nucleus, except camel, llama. So camel and llama, these are mammals, but they having nucleated RBC, oval shape RBC. Oval shape nucleated RBC present in this camel and llama. Okay. So I hope you understood this concept. Next. So WBC, very important. So the lifespan of WBC 3 to 7 days, some book 13 days it is given, but NCERT it is not given. But the short duration, life cycle is very short period of time, just you remember. So, right? The short period of time they can survive. So they are nucleated cells. Most abundant cells of the blood are nucleated or enucleated. They will ask you question. The most abundant cells of the blood are enucleated or without nucleus. Now, based on presence or absence of granules in cytoplasm of the WBC, we classified them into granulocyte, agranulocyte, with granules in cytoplasm, without granule in cytoplasm. Okay. So why we are calling WBC? Because lack of hemoglobin pigment. There is no hemoglobin pigment, because of that we are calling them WBC, white in color. Now granulocytes are three types: basophil, eosinophil, also called acidophils and neutrophils. Acidophils and neutrophils. Right? So basophils, their number, the total number of basophil 0.5 to 1% to the total WBC. Right? So 2 to 3% eosinophil, 60 to 65% neutrophils, 6 to 8% monocyte, lymphocyte 20 to 25%. These are very, very important. The total number of WBC, right? In the total number of WBC, 2 to 3% are eosinophil, 0.5 to 1% is basophil, 60%. Most abundant cells of the WBC are neutrophils. Least abundant cells are basophils. Okay. So after neutrophil, most abundant cells are lymphocyte. Okay. So agranulocyte, they are also formed in bone marrow. Granulocyte, they also form in bone marrow. The function you can see. Basophil, the nucleus you can see 2 to 3 lobes or S-shaped nucleus present. The nucleus of the basophil S-shape are 3 lobes. 3-lobe nucleus present. 3 lobes are present. 3 lobes. They secrete heparin, histamine, serotonin. Right? So they produce inflammatory reaction. You know heparin function. Right? Histamine, serotonin, vasoconstrictor, vasodilator, you may knowing that. So they produce inflammatory reaction. Next one, eosinophil, bilobed nucleus present. Bilobe. 2 lobes are present in the nucleus. So the number eosinophil, you can see they resist infection. They can fight with infection and they try to eliminate the infection. They resist infection and their number will increase during parasitic infection, allergic reaction. Allergy is caused because of the basophil. Basophils are involved in allergic reaction. Very important. Immunology chapter we studied. Whenever an pollen grain enter into our body, basophils are activated and they release histamine, serotonin, right? They are responsible for inflammation, right? But here, the production of these eosinophil number will increase during the allergic reaction. That is very, very important. Next, neutrophil. Neutrophils are phagocytic cells. They can engulf bacteria, pathogen, they can destroy or they can eliminate. Right? Now you can see basophil, eosinophil, neutrophils. Together we are calling polymorphonuclear leukocyte. Polymorphonuclear leukocyte because the nucleus shape is different. The shape of the nucleus is different. You can say here the lobes are, many lobes are present in the nucleus. Very, very important. There are many lobes are present in the nucleus of the neutrophil. So we are calling polymorphonuclear leukocyte. Right? These are all also known as a polymorphonuclear leukocyte. Not only this one. Lymphocyte, after that, next one agranulocyte, monocyte and lymphocyte. These together forming agranulocyte. Right? Among the lymphocyte, among these agranulocyte, monocytes are phagocytic cell. They converted into macrophages. The nucleus shape, kidney shape, reniform shape or kidney shape. Okay, kidney shape nucleus present. Here round shape or spherical shape nucleus present in lymphocyte. These are the smallest cells of the WBC. Macrophages or monocytes are largest cells of the WBC, largest cell. So these lymphocytes are again classified into B lymphocyte, T lymphocyte. The lymphocyte produce a group of protein we are calling antibodies. So that antibodies helps in production of immunity, or they can eliminate pathogen or antigen that enter into our body. Right? Next one. So here one more point you remember here. All clotting proteins or clotting factors are present in plasma in inactive condition. So all clotting factors are inactive condition. In active condition. Very, very important. So clotting factors present in plasma in inactive condition. Right? Only in inactive condition. Okay. So this is about the WBC. 1 minute. Give me. Next one. Platelets. So I told you platelets also called thrombocyte, and here one more important point, the number is 6000 to 8000 per cubic milliliter of the blood. 6000 to 8000 number cubic milliliter of blood, very, very important. So thrombocytes also called platelets. Now the formation of the thrombocyte is different from the formation of the WBC and RBC. You can say this is megakaryocyte which present in red bone marrow. Megakaryocyte. Mega means large nucleus. Karyon means nucleus. Megakaryocyte. These are the cells that present in red bone marrow. Red bone marrow. Okay. So now these megakaryocyte undergoing fragmentation. It will undergo fragmentation and forming smaller fragments. Fragmentation. Breaking. Okay. So now all these we are calling platelets. So now platelets are forming by breakdown of the megakaryocyte into smaller and smaller pieces. Right? So platelets are very, very important. They secrete some factors. They release certain factors. So these certain factors are very much needed for the clotting of the blood. Example, thromboplastin, tissue factor. So that is released by the platelets. If decrease in the number of platelets, that lead to clotting disorders. So the average number healthy person 1,50,000 to 3,50,000 per cubic milliliter of blood. So cubic milliliter of blood contain 1,50,000 to 3,50,000 of platelets or thrombocytes. Right? When the number of platelet decreasing means that lead to clotting disorder, because platelet releases certain factors that help in clotting of blood. Okay. So this is the second most highest abundant cells in blood after RBC. Right? So arrange the following blood cells or formed elements based on their number. RBC 5 lakhs, followed by platelets, followed by WBC. So ascending order, descending order based on their count, number. So they will ask you question that you can remember. Right, this is the question. So erythropoiesis starts in. So this is a tricky question. Example, liver, I told you, erythropoiesis takes place in liver, spleen, red bone marrow, kidney. So they are asking in which stage erythropoiesis starts, when we are in embryonic stage, Erythropoiesis starts in spleen, liver, and also told you yolk sac. Yolk sac also helps in production of WBC. Right? Erythropoiesis starts in spleen. Okay? During early embryonic stage. Liver and yolk sac. Next one, name the blood cells whose reduction in their number lead to clotting disorder. Excessive loss of blood from the body. That's simple. I told you thrombocyte. Right, serum differ from blood in lacking. This is 2016 phase 1, phase 2. Serum differ from blood in lacking. So I told you clotting factors will absent. Right? So clotting, not albumin, not globulin, not antibodies, but clotting factor, blood minus or plasma minus clotting factor, you will get serum. Right? So option that one. So matching question. So very important. It is given in 2020 phase 2 corona time. So lymphocytes immune response because they produce antibodies. Neutrophils are phagocytic cells. Okay. Eosinophil, right? Destruction, release histaminase. Basophil release granules contain histamine. This is the question I told you. Eosinophil number will increase during the allergic reaction. Basophil number or basophils are responsible for allergy. So basophil release histamine. Now when basophils are releasing histamine, that can be responsible for causing allergy. When allergy is there in the body, eosinophil number will increase because they can release histaminase enzyme, right? Destructive enzyme that is released by the eosinophil. So now that is the answer based on that. So there are blood grouping system. So NCERT line, I will be explaining this about this blood grouping system. 1 minute. Right. So the human blood is differ in certain aspect, although it is similar. This sentence is also very important. The blood of the human beings differ in certain aspect, although it is a similar. Right? So there are various blood groups are present, but throughout the world there are two blood groups are majorly accepted. Right? So these are all NCERT line you should remember. The blood differ in certain aspect, although but it is same. Okay, there are various types of blood group have been described, but there are two blood grouping system we are using throughout the world. Throughout the world important. The two blood grouping system are ABO blood grouping system and RH blood grouping system. These two blood grouping systems are used throughout the world. So what is the basis for this formation of ABO blood grouping system and RH blood grouping system? So right, the formation of this ABO blood grouping system because of presence or absence of antigens, antibodies. Antigens, antibodies. Antigen is a chemical which is responsible for inducing immune responses. Right? So antigen is a chemical, it is responsible for inducing immune response in the body. That is called antigen. So that antigen may be a polysaccharide, or it may be a protein, or it may be a lipid, or it is maybe a nucleoprotein, or nucleic acid. So whenever we are exposing to certain antigen from outside, our body produce certain response. Our group of proteins are released by our body against those substances. We are calling antibodies there. So what is antigen here? Antigen that is stimulating, that is inducing immune response in our body. That is called antigen. So that antigen is a chemical that may be a polysaccharide, maybe a protein, or maybe a lipid, nucleoprotein, or nucleic acid. But antibodies are always proteins. They are always proteins. The group of proteins produced by our body or immune system against these antigens. The group of proteins or army of proteins produced by our immune system against these antigens. Against the antigen, there is a group of proteins are produced in our body. So that group of proteins we are calling antibodies. So question important. Antibodies are always protein, but antigen may be a polysaccharide or a protein or a lipid or nucleoprotein, nucleic acid. Right? So there are two types of antigen present on RBC. Antigen A, antigen B. There are two types of antigen present on surface of RBC. RBC surface, it contain two types of antigen, either antigen A, antigen B. The two individuals, the plasma of the two different individuals contain two different antibodies. Two different antibodies are present in the individuals, two different individuals. Two different antibodies are present in two different individuals. Example, antibody A, antibody B. Antibody A, antibody B. So antibodies are present in plasma, antigens are present on the surface of the RBC. Okay, so this is very, very important. So the presence or absence of antigen or antibody on RBC in plasma. Because of that, we are getting ABO blood grouping system. ABO blood grouping system. Right? So A blood group person on surface of RBC, antigen A present. This is antigen A. In plasma, antibody B present. In B blood group, on surface of RBC, antigen B present. In plasma, antibody A present. In ABO blood group person, on surface of RBC, antigen A, antigen B, both are present, but there are no antibodies in plasma. Antibodies are absent. O blood group person, no antigen on surface of RBC, but plasma contain antibody A, antibody B. Okay, so how can you differentiate A blood group from B blood group? A blood group person on his RBC, antigen A present, in his plasma, antibody B present. B blood group person on his RBC, antigen B present, in plasma, antibody A present. AB blood group person, antigen A, antigen B, both are present on RBC, but no antibodies in plasma. O blood group person, no antigens on RBC, but anti-A, anti-B antibodies are present in plasma. Okay, so now donor group I will explain that. Whom can they donate? This is very, very important. So O blood group can donate blood to the AB. O can donate blood to the A. O can donate blood to the B. A can donate blood to the A. B can also donate blood to the AB. Now, but B can receive blood from the B and also O. A can receive blood from the O and also from A. But O can receive blood from only A. O can receive blood from only A. B can receive blood from O, A, B and AB. So this is universal donor recipient, and this is universal donor, right? So O negative, AB positive, universal donor, universal recipient. Very, very important. This chart is important. So based on these also, you can remember where is whom can they donate the blood, whom cannot, right? So this is very, very important. One next, RH grouping. RH blood grouping. So actually one more antigen beside earlier I said there is only antigen A, antigen B on the surface of RBC. But later one more antigen, it was first discovered on rhesus monkey on the surface of RBC of rhesus monkey, along with antigen A, antigen B, one more antigen also discovered. So it was discovered in rhesus monkey first, so we are calling this RH antigen. RH antigen. If these RH antigen present on RBC, that persons are known as a positive person. If there is no RH antigen, the person is called RH negative person. So 80% populations RH positive, only 20% of the populations are RH negative. Okay, so any person if RH antigen present in on is RBC is a positive. If there is no RH antigen on his RBC, is a negative person, right? So how many, sometimes they will ask you simple question. So how many antigens present on AB positive blood group person? Then the answer will be 3. Antigen A present, antigen B present, and one more RH antigen also present. So on the surface of the AB positive person, there are two antibodies are present. Sorry, 3 antibodies are not antibodies, antigens. 3 antigens are present. These kind of question they will ask you. A positive person, how many antigens are present? A positive person, antigen A present, RH antigen 2, positive person. Negative person contain only 1 antigen A. O blood group person, how many? O positive person, how many RH antigens are present? There is no antigen A, there is no antigen B on RBC of the O blood group person. If it is positive, only RH antigen present on RBC. Antigen A, antigen B absent. Okay, so they will ask you this kind of question, you remember. So erythroblastosis fetalis, you know, it is a condition where the marriages between father RH positive, mother RH negative, and developing fetus is RH positive. In this condition, you can see this is the blood. Both the blood, fetal blood and maternal blood, both are separated by placenta. This is the placenta. Placenta. Now you can see mother is RH negative. These are RBC. Mother is RH negative. So on RBC surface, there is no RH. Here, no RH antigen. RH antigen absent. Mother's blood. Mother's RBC does not contain any RH antigen because she is negative. No RH antigen. But fetus blood, you can see this is the fetal blood cells, right? So on the surface of the fetal blood cell, just I am drawing, you can see white color, these are Rh antigens. So Rh antigens are present, positive, so Rh antigen present on fetal RBC surface. So during the first delivery, you can see there is a rupturing, there is a breaking of the placenta, there is a, you can see there is mother blood, chance of mixing maternal blood with fetal blood. So then, so RBC which contain Rh that may enter into mother's blood, and the RBC from the mother also enter into fetus. But mother blood doesn't have any Rh antigen on their surface, so nothing will happen here. But here you can see, originally the mother RBC doesn't have any Rh antigen, but you can see this is the RBC of the fetus with Rh antigen. So moving into mother's immune system recognizing this as a foreign body. So mother's immune system recognizing this Rh antigen as a foreign body or antigen, and mother immune system producing IgM antibody first time. IgM antibody. So IgM antibody is a large antibody that cannot cross the placenta, right? So whenever we are exposed to the pathogen, first antibody that will be produced our body by IgM. Later it will convert it into IgG. So now first delivery, before this IgM reaching to the fetus, the first delivery will be the same. Safe, nothing will happen to the first delivery. Suppose same lady second time she conceived. Second time she conceived. So the fetus again positive fetus. Already the antibodies are present in mother. You can see. Already there are antibodies are already produced against that Rh antigen. So now these Rh antigen, they cross the placenta and enter into the fetus. They can destroy RBC, right? So the baby will suffer with jaundice or destruction of RBC, so it lead to anemia, jaundice. Sometime it lead to death also, right? So I hope you understood RH concept. Very important. Remember. Okay. So this is type of open type of circulatory system. Open type of circulatory system, closed type of circulatory system. So already you studied, but this is very important. Careful. Open type, you know. Example you should remember. Arthropoda, non-cephalopod mollusca, very important. Example, octopus, squid, laligo, devilfish, cuttlefish. These are all examples of non-cephalopod mollusca. Echinodermata, hemichordata. These are all invertebrates. Okay. So here cephalopod mollusca example, octopus. Here non-cephalopod. So non-cephalopod mollusca, echinodermata, hemichordata, arthropoda. Right? These are all invertebrate with open type circulatory system. But urochordata is the chordata with open type circulatory system. This is chordata, urochordata, right? Now you can see closed type circulatory system, blood present in blood vessels. The diameter of the different blood vessel is varying. So Annelida, cephalopod mollusca that I told you, octopus. So non-cephalopod, cephalopod. Cephalopod example, octopus. And cephalochordata, vertebrata. These two chordates, these two invertebrates. So question they will ask you, invertebrate phyla with open type circulatory system, invertebrate phyla with closed type circulatory system. chordate with open type circulatory system, or vertebrate with open type circulatory system, or chordate with open type circulatory system or closed type circulatory system, they will ask you. Or they will ask you the examples of belongs to this phylum. Example that belongs to the phyla also they will give you, and they will ask you. Identify one example. Invertebrate phyla. Example of invertebrate phyla with open type circulatory system. Okay. So this is very, very important. Right. Lymphatic system. Lymphatic system, when blood flowing through the capillaries, when blood passes through the capillaries. Okay, these are capillaries, these are all venules. Okay, now you can see when blood, right, passing through the capillaries, these are capillaries, right, water and many water soluble substances, they are leaking into the cells that present in the tissue. You can see these are all the cells that present in the tissue. This is the tissue. Okay, now the gap or the space between the cells we are calling interstitial space or intracellular intercellular space. Not interstitial space or intercellular space. Now when blood passes through the capillaries, water along with many water soluble substances, right, so they'll enter into the cells, enter into the interstitial space or gap, and leaving here only large plasma proteins, they present inside the blood only, and most of the formed elements except lymphocyte. Most of the formed elements and large plasma protein, they present inside the blood, but water and water soluble substances, they enter into the gap or the space that present between the cells of the tissue. We are calling interstitial space. Now the liquid which is coming from the plasma into the interstitial space, we are calling tissue fluid. Tissue fluid or interstitial fluid. Interstitial fluid. So this interstitial fluid contain many water soluble substances and water, right? But except these are absent. Now the mineral distribution of the tissue fluid or interstitial fluid and plasma same. The mineral composition of the tissue fluid and plasma both are same. The main function of this tissue fluid that helps in exchange of the material, right? So nutrients, exchange of the nutrients and gases between blood and cells, between blood and tissue, by only this fluid. Okay, the exchange of the material, that is nutrients, nutrient and gases, oxygen and carbon dioxide, always occur between the cells and the tissue. The tissue is called tissue fluid. Interstitial fluid or liquid. The interstitial fluid is a liquid which mainly helps in transportation of nutrient and transportation of gases between the tissue and blood. Okay, right. That is called tissue fluid. Next one. Now there is an elaborated network of blood vessels that present this. You can see among these there is an elaborated network of the blood vessels. You can see there is an elaborated network of the blood vessels. So these elaborate, I think where I will take another color, yellow color. You can see this is an elaborated network of blood vessels that present in between the tissue. These elaborated network of blood vessels we are calling lymphatic vessels. Lymphatic vessels. Okay, lymphatic vessels. Now the tissue fluid from the tissue that is collected into lymphatic vessels. The tissue fluid which is collected into lymphatic vessel we are calling lymph. Okay, lymph. So lymph is collected from the tissue. What is lymph? The interstitial fluid or the tissue fluid which is enter into a network of blood vessel that is called lymphatic vessels. Once the tissue fluid enter into lymphatic vessel we are calling it lymph. So now this lymph is drained into major vein. Drained into major vein. Simple. Suppose from here 100 ml of blood is entering into the capillaries. From these 100 ml of blood, 15 ml of blood is leaking into the tissue. Only 85 ml is collected into vein. Again this 55 ml is collected by the lymphatic system into draining into major vein. Okay, this is about lymph. So lymph is a colorless fluid. So it present in lymphatic system. It contains specialized cells called lymphocytes. They're responsible for immune response because they produce antibodies. Very important. So lymph is also important carrier of the nutrients, hormones, and helps in absorption of the fat through the lacteal present in villi, microvilli, villi of the small intestine, villi of the small intestine. Okay. Next, coagulation or clotting mechanism, very important, right? So this only this chart you remember, you will any question you will get, you can answer, you able to answer any question if you remember this chart. Simple. Whenever there is an injury or trauma, coagulation means simple liquid part of the blood. So whenever there is an injury in the body, the liquid part of the blood is coming out through the injured part. So the prevention of the liquid part of the blood into solid part and preventing the loss of blood from the body, that is called coagulation, right? So coagulation prevent loss of blood from the body through the injured part. So this coagulation mechanism is activated by injury or trauma, or at the site of damaged tissue, injury site. This is the site of injury. At the site of injury, there is a tissue will be damaged. The damaged tissue at the site of injury are platelets that present in the blood activates or they release certain factors. They release certain factors. That factors NCERT not mentioned. That factor is called thromboplastin or tissue factor. This is the third factor, right? So this thromboplastin activating inactive thrombokinase enzyme, that is 10th factor. Inactive thrombokinase is an enzyme that present in the blood, but it is inactive condition. When thromboplastin is formed, this thromboplastin is converting this inactive thrombokinase into active thrombokinase. So formation of this thrombokinase is a complex enzyme. Complex enzyme, it required cascade of reaction. It is formation of this thrombokinase is a cascade of reaction. So one reaction can activate other reaction can activate another reaction, and finally we will get active form of thrombokinase. That is called cascade of reaction. So once we will get this active thrombokinase, that active thrombokinase helps in prothrombin. Prothrombin also clotting protein, I told you they present in inactive condition. So there is prothrombin converted into thrombin, active, right? So once thrombin is formed, fibrinogen is a soluble protein that present in plasma. But once thrombin is there, that thrombin convert this fibrinogen into fibrin. So fibrin insoluble, and that fibrin will form fibrin threads, right? So in these fibrin threads, a dark color, reddish dark color scam will form, right? So this scam where all formed element, blood and platelet, WBC, they will stuck and they will prevent loss of blood through the injured part. Okay, so this is very, very important. So here erythroblastosis fetalis condition arises when, so father is female, RH negative, fetus RH positive first time, right? Female RH carrier, fetus second time. Condition very, very important. Okay, so already I told you. So how they will ask you question, you can see they will ask you like this, right? So blood vascular system. So the blood human circulatory system is also called vascular system, also known as vascular system. It including three parts, or it includes three parts. One, the liquid part, blood already we completed. Next one, the muscular heart and blood vessel, arteries, vein and capillaries. These are the parts. So mesodermal in origin, location is thoracic region between two lungs. There is a gap called mediastinum. So thighs clenched fist, the size is clenched fist. So heart is protected by the pericardium is a layer, it is a pericardium layer. So outermost layer pericardium, right? The wall of the heart is contain three layers, right? Innermost layer you can see endocardium, and thickest layer is called myocardium, outer is called pericardium. Outside this is pericardium, outside pericardium layer, myocardium, endocardium, not this is pericardium, this is epicardium. Epi wall contain three layers, endocardium, myocardium, epicardium. So epicardium outside there is a pericardium present. So outer fibrous pericardium, inner serous pericardium. Serous again divided into visceral pericardium and parietal pericardium. That is not given, just you remember, right? So next one, this is important. So the main important points I will tell you. So four chamber heart, you know, the upper chambers are smaller chambers, lower chambers are ventricles, larger chambers, pumping chambers, and these are receiving chambers, right? Now here you size, you can see the increasing size. Left ventricle is the largest one, or thick layers, thick muscles are present, followed by right ventricle, right atrium, left atrium, the sequence. Size, okay, you can see this is the thickest wall, larger chamber, followed by right ventricle, right atrium, left ventricle. Okay, so ventricles are thick compared to atria. Ventricles are thick muscles compared to atria, right? Right, you can see. So right atrium, left atrium, both are internally separated by a septum called interatrial septum. Interatrial septum, this is thin, right? So left and right ventricle also separated by one more septum. This is also, this is thin and this is thick. This is called the interventricular septum. Right ventricle, left ventricle, there is a structure called interventricular structure. uh, both the chambers are separated by the interventricular septum, which is thick fibrous, thick. Now upper chambers and lower chambers are separated by thick fibrous septum. Very, very important. Thick fibrous septum, right? So this atrioventricular septum having openings. So the atrioventricular septum having openings, small openings are present. The small openings that present in atrioventricular septum we are calling right atrioventricular aperture, left atrioventricular aperture. Now you can see right atrium open into right ventricle. So through the small opening that is called right atrioventricular aperture. So it is guarded by tricuspid valve. Tricuspid valve. Okay, right atrium open into right ventricle. So right ventricle open into pulmonary artery. Pulmonary artery. So that supplies the blood to the lungs. Pulmonary artery. So the right atrium opening into pulmonary artery, here it is guarded by semilunar valve. Pulmonary artery open into lungs, right? From the lungs, oxygenated blood collected by the 4 pulmonary veins. Pulmonary veins are opening into left atrium. So left atrium open into left ventricle through the left atrioventricular aperture. So guarded by bicuspid valve, also called mitral valve, right? So left ventricle open into aorta. Here also it is protected by semilunar valve. Aorta pumping blood, oxygenated blood to the body parts. Again, from the body parts, the blood is collected by vena cava. Superior vena cava, inferior vena cava, coronary sulcus, collecting the blood from the various parts of the body, and they open into right atrium. Okay. Now tricuspid valve, bicuspid valves are also called AV valves because they present between atrioventricular aperture or septum. AV valves, right? So conducting system of the heart. Entire heart is made up of with cardiac musculature. Entire heart is made up of with cardiac musculature. A specialized cardiac musculature that present in the heart we are calling nodal tissue. Nodal tissue is a specialized cardiac musculature that is distributed in the heart we are calling nodal tissue. The patch of these nodal tissue which present in upper right corner of the right atrium. The patch of the nodal tissue which present in upper corner of the right atrium. Upper corner of the right atrium we are calling SA node, sinoatrial node. Now here a nodal tissue has the ability to generate action potential without any external stimulus. That's why this is acting as an auto-excitable, right? Auto-excitable, no need to give any stimulus from externals. Itself it can generate action potential without any external stimulus. So nodal tissue has the ability to generate action potential without any external stimulus. So it is a auto-excitable. Now these patch of the nodal tissue which present in right atrium, upper corner of the right atrium we are calling SA node. One more patch of this nodal tissue which present left corner of the right atrium, closer to the interventricular septum. We are calling AV node. Atrioventricular node or AV node. Right? This AV node, so coming out through the atrioventricular septum, emerging on interventricular septum, top of the interventricular septum, dividing immediately into right branch, left branch, and each branch dividing into small fibers we are calling Purkinje fibers. All that Purkinje fibers are connected to ventricular musculature, right? So this is conducting system. SA node, AB node, bundle up is bundle branches, Purkinje fiber. Okay. So now the generation of action potential by these parts is not same, it is various. SA node can generate 70 to 75, right? The number of action potential per minute generated by these conducting parts are not same. SA node can generate 70 to 75 action potential per minute, so that's why it is called pacemaker. Pacemaker. AB node generate 40 to 60 action potential per minute. So there is remaining parts can generate 20 to 30 per minute. Okay, conducting system. This is the conducting system. SA node, AB node, bundle up is bundle branches, Purkinje fiber. Okay. Cardiac cycle. So cardiac cycle, generally cardiac cycle is completed in 0.8 second, in healthy individuals 0.8 seconds. So that 0.8 second, atrial systole 0.1 second, ventricular systole completed in 0.3 seconds, and giant diastole completed in 0.4 seconds. Okay. So the event, what is actually systole, what is actually diastole, I will tell you. Relaxation of any chamber we are calling diastole, or repolarization. Repolarization or relaxation. Relaxation of any chamber we are calling diastole or repolarization. Contraction of any chamber we are calling systole or depolarization. Depolarization. So sometimes some book depolarization lead to atrial systole. That is also right. Okay. So atrial systole, ventricular systole, giant diastole. So these are the events that takes place in cardiac cycle. Okay. So alternate contraction and relaxation of atria and ventricle together we are calling heartbeat. So the sequential event from beginning of one heartbeat to the beginning of the next heartbeat we are calling cardiac cycle. Now you can see before atrial systole, that is the first event. For the four chambers, now they present in giant diastole condition. That means the four chambers are in relaxed state. So that time tricuspid valve, bicuspid valves are also known as AV valves are open. Semilunar valves are closed. Semilunar valves are closed. You can see. Okay, tricuspid valve, bicuspid valves are open. Semilunar valves are closed. You can see semilunar valves are closed. Semilunar valves are closed, tricuspid, bicuspid valves are open. Now, vena cava, you can see inferior vena cava, superior vena cava, and coronary sinus collecting blood into right atrium. Pulmonary veins from the lungs collecting blood into left atrium. Right? Now, from the left atrium, the blood enter into left ventricle. From the right atrium, blood enter into right ventricle. Okay, right ventricle. So now you can see during the giant diastole, 70% of the ventricles are filled. The 70% of the ventricles are filled during the giant diastole. right? Now, what happens now? SA node generating action potential. That action potential passing to the right atrium and also left atrium. Passing to the right atrium and left atrium. Both right atrium and left atrium. So already just imagine already 70% of the blood already filled with 70% of ventricles already filled with blood because of giant diastole. Now you can see SA node generating impulse, right? The blood is present in the right atrium and left atrium, right? When the ventricles are filled with blood, that creating pressure on AV valves. Now that AV valves are open, because of blood enter into right and left atria, the pressure inside the wall of the right atrium and left atrium is increasing. So now they are undergoing contraction. They are undergoing contraction. When this right and left atria undergoing contraction, we can say it is a atrial depolarization, atrial systole, atrial contraction. Because of this atrial contraction, ventricles are filling only 30%. So 70% is already filled. Only 30% of the ventricles are filled during the atrial systole. Right? At the end of the atrial systole, also end of the ventricular diastole. Right? So at the end of the atrial systole, also end of the ventricular diastole. Now the total amount of blood that present in ventricle at the end of the ventricular systole, sorry, atrial systole or ventricular diastole is called end diastolic volume, that is 120 ml. In 120 ml, 84 ml already is there. Only 36 ml is coming. This is 30%. This is 70%. Okay, already 84 ml of blood already there in ventricle, but when atria undergoing contraction, 36 ml of blood enter into respective ventricles. So the total amount of blood at the end of the atrial systole is 120 ml, right? So that is called end diastolic volume. It is a atrial systole or contraction. Why we are calling end diastolic volume? When atria undergoing contraction, at the same time ventricles are undergoing diastole. So that's why we are calling end diastolic volume, right? Sorry. Next. Now, so the blood is present in pressure increasing in the ventricles. When pressure is increasing in ventricles, right? So slowly tricuspid wall and bicuspid walls are closing. Pressure in ventricle slowly increasing. You can see the blood pressure in ventricle is increasing. When pressure increasing, here less and here more pressure. So then there is a chance of backward movement of blood from the atria to ventricle to atria. To overcome that problem, tricuspid wall, bicuspid wall, they are closing, or AV valves are closed. They produce first sound of the heart, lup, lup, lup. Okay, lup sound will produce, lup sound, lup sound. Right? Now, slowly you can see the pressure is increased in ventricle and the pressure is less. The pressure of the blood in aorta, respective aorta, the blood pressure is less. So blood from the ventricle, now ventricles are undergoing contraction. Ventricles are undergoing contraction, right? So what is the total amount of blood that left here? 120 ml. Now each ventricle sending 70 ml of blood into respective aorta. When ventricles are undergoing contraction, 70% of the blood, so that is 70 ml of blood enter into respective aorta, systemic aorta, pulmonary aorta, right? So that enter into this aorta, right? So now at the end of the atrial systole, the total amount of blood that left in ventricle. So now you can see it is sending 70 ml. So minus 70 ml, that is 50 ml. So at the end of the ventricular systole, the total amount of blood which is left in both the ventricle is 50 ml, 50 ml, 50 ml. So this is called end systolic volume. End of the ventricular systole. At the end of the ventricular systole, the total amount of blood left in both the ventricle is called end systolic volume. So end diastolic volume minus end systolic volume, you will get stroke volume. So 120 ml minus 70 minus 50, you will get 70 ml. Now you can see stroke value means the amount of blood pumped by the both the ventricle into respective aorta per minute. That is stroke value 70 ml, right? In giant diastole, again slowly the pressure in ventricle decreasing. When ventricular pressure is decreasing, there is again chance of backward flow of blood from the respective aorta. So semilunar valves are closing, producing second sound, right? So now again there is a decreasing in the pressure in ventricle. So tricuspid, bicuspid valves are open. Again the first two I told you giant diastole, that will be repeated. Okay. So the giant diastole. So the volume of blood ejected out by the ventricle per beat is 70 ml. That is important. So cardiac output. So cardiac output is equal to cardiac output, the amount of blood pumped by the left and right ventricle per each minute. Per each minute important. So cardiac output is equal to stroke volume into heart rate. Right? So stroke volume is 70 ml, number of heartbeats, average 72 or 70 to 75. Right? The stroke volume is 5.2 liter. If you take 72 beat, it is 5.40 liter. Okay, important, they will ask the question. ECG, next one, electrocardiogram, electrocardiograph. Electrocardiograph means is a device. Graph means is not a graph paper, remember. Electrocardiogram means is a device or machine. Electrocardiogram means this is the paper, right? That image that we can see on monitor of the screen, or we can print on the paper, that is called cardiogram. And electrocardiogram is a device which we are using to record this electrocardiogram. Okay, so right, in a typical electrocardiogram we use 3 electrical leads to record continuous recording. For continuous recording of the ECG we need 3 electrical leads, right? For complete evaluation of ECG we need 12 electrical leads. 12 electrical leads we will using to continue for complete evaluation of heart functioning. But here we are using 3 electrical leads which is connected to the each wrist and left ankle for continuous monitoring of the heart functioning. Important, right? So in ECG you can see there will be each peak is denoted with letters P, Q, R, S and T. Here P wave indicates that depolarization of atria and atria begin to contract, right? QRS complex is indicating ventricular depolarization or ventricle begin to contract shortly after Q. T wave representing that ventricles are coming to their original position, that is repolarization, giant diastole, that is diastole. But here atrial depolarization is not apparent or not recorded, but it is recorded also, it is masked by QRS complex. QRS complex. Okay. Hepatic portal circulation, there is a special connection of the blood vessel between liver and small intestine, we are calling hepatic portal circulation. Coronary circulation is a special type of blood vessel that's supplying. There are 2 coronary arteries from the systemic aorta arising and submitting or supplying blood to the heart muscles. That is coronary circulation. So blood vessels are, there are arteries, veins, venules, right, are there. So you remember, because of systemic circulation supplying nutrition, oxygen, various component to the body cell, and also helps in elimination of carbon dioxide waste material. That is the function of systemic circulation. Right here, blood vessel, we are taking arteries and blood. The blood vessels are surrounded by three layers: tunica, intima, tunica media, sorry, and tunica externa. So these are the three layers that present in blood vessels, vein or artery. Three. These are three present in vein and artery. Tunica intima, outer layer, innermost layer, made up of its squamous endothelium, simple squamous epithelium, that is called endothelium. Simple squamous epithelium called endothelium, and this is made up of its smooth muscle and elastic fibers. Elastic fibers are present. Very, very important. Smooth muscle, elastic fiber. Outermost layer is fibrous tissue, fibrous connective tissue, and collagen fibers. So tunica externa is made up of with the fibrous connective tissue and collagen fibers. It is made up of with smooth muscles and elastin fibers, and innermost is made up of with squamous epithelium, that is endothelium. Now here, tunica media is thick in arteries and thin in vein. Thin and thick, very, very important. So these are the blood vessels. So right, so regulation of cardiac function. So generally it is neural system and chemicals, hormonal and also neural system that can regulate the functioning of the heart. But it is a myogenic heart because it can regulated by the special type muscle that present in heart, that is called nodal tissue. So it is auto-regulated, but there is a center called hormones also regulating the function neural system. Medulla oblongata, there is a center present. That center control the functioning of the heart. Cardiac center. Cardiac center that present in medulla oblongata controls the functioning of the heart. Slow beating or fast beating. Autonomic nervous system also controlling the functioning of the heart. Sympathetic nervous system increasing the heart rate and cardiac output. And same parasympathetic nervous system of the autonomic nervous system, ANS, decreasing the heart rate, thereby cardiac output, right? So hormones also controls, example, adrenal medullary hormones, that is catecholamine hormones, epinephrine, norepinephrine, they can increase the function of the heart. Increase the function of the heart, that is very, very important, right? This is sympathetic and parasympathetic. So disorders of heart, hypertension. So the repeated BP checkup, if that reaching 140/90 frequently. So the repeated checkups of the BP of a person, if it is reaching more than 140/90, we can say it is a hypertension. So hypertension lead to the damage to the vital organs like brain, kidney. So it can high BP can damage vital organs of the body like brain, kidneys, right, and also heart, right? Coronary heart disease, this is very, very important. Coronary artery disease, CAD, because of the calcium, right? So lipids, calcium, fibrous tissue, these are all forming a flake in the blood vessel that decrease the size of the lumen of the right blood vessel that is supplying blood to the heart muscles. So that lead to disorder called coronary heart disease. Insufficient supply of the blood to the muscles or insufficient supply of the oxygen is reaching because of the narrow lumen. because of this calcium deposition and fibrous tissue and cellular deficit. So CAD also called atherosclerosis. Atherosclerosis. Angina. So enough oxygen is not reaching to the heart muscle. Sufficient or enough or sufficient blood with oxygen is not reaching to the cardiomyocyte of the heart. So that lead to angina pectoris. Acute chest pain is the symptom of the angina. Acute chest pain. Acute chest pain is the symptom of the angina pectoris. Very, very important. Due to reason is enough oxygen is not reaching, enough blood is not reaching to the myocyte. Now the person, any age, men and women of any age can suffer with these disorders, but most common in elderly aged people, old age people, women, elderly aged and aged women, elderly. Heart failure and heart attack, cardiac arrest, these are very, very important. Heart failure means heart stops pumping, heart stops pumping. Okay. Heart attack means damage. Okay. Heart failure. Heart attack means damage to the muscle cells. Right. Suddenly damage of the muscle cell due to myocardial ischemia. Right. Heart attack also called, it is myocardial infarction. Myocardial infarction. Damage of the tissue. Cardiac arrest means heart stops pumping, heart stops beating, or heart stops working. Cardiac arrest. Heart failure. Right? Unable to supply the needs of the body. Needs of the body. The heart is unable to pumping the blood, the needs of the body. That is called heart failure. Cardiac arrest means heart stops beating. Heart, heart stops pumping. Heart stops beating. Heart stops beating. That is cardiac arrest. Okay. So this is all for today. So I explained briefly line to line of NCERT, most of the important important point. So that will I covered that most expected question from those areas I covered in this chapter. So I hope this will helpful for you, and keep concentration on the videos and read NCERT line to line and revise NCERT line to line daily 2 hours. So that will helpful for you to crack 340 in biology in NEET exam 2024, right? So thank you all. Good night.